Hello everyone. In this lecture, we are going to discuss protein translations. Since we get some basic knowledge about the gene expression, transcriptions, and gene replications. So this lecture will mainly focus on how mRNA can produce proteins. Translation proceeds in three phases. The initiation, the ribosome assembles around the target mRNA. The first tRNA is attached at the start codon. The initiation, the tRNA transfers an amino acid to the tRNA corresponding to the next codon. The ribosome then moves to the next mRNA codon to continue this process, creating the amino acid chain. The termination, when the poly-tRNA encounters a stop codon, and then the ribosomes fold the polypeptides into its final structure. There are several key components for protein translation. The mRNA is the template and information counters for protein translation. Amino acids are building block carrying an amino acid to the protein synthetic machinery. ATP and GTP supplies energy. Ribosome serves as the site of protein synthesis. The protein complex plays roles as initiation factor, negation factors, and termination factors. A transfer RNA molecule is used in translation and consist of a single RNA strand that is only about 80 nucleotides long. The tRNA carry an amino acid to ribosome as directed by a 3-nucleotide sequence in the messaging RNA. The anticodon is a unit made up of 3 nucleotides that correspond to these 3 bases of the codon on the mRNA. The structure of the tRNA can be decomposed into its primary structure, its secondary structure, and its tertiary structure. The clover leaf structure becomes the 3D L-shaped structure through coaxial stacking of the helix. The tRNA structure consists of the following. A 5-terminal is the acetyl stem is a 7 to 9 base pairs stem made by the base pairing of the 5' terminal nucleotides with the 3-terminal nucleotide, which contains the CCA 3' terminal group used to attach the amino acid. In general, such 3-terminal tRNA-like structures are referred to as genomic tags. The CCA tails is a cytosine-adenine sequence at the 3 end of the tRNA molecule. The amino acid located onto the tRNA by amino acid tRNA synthesis. To form amino acid tRNA complex is covalently bound to the 3-hydrogen group on the CCA tails. This sequence is important for recognition of tRNA by enzymes and critical in translation. The D-arm is a 4 to 6 base pair stem ending in a loop that often contains dihydrouridine. The undercodon arm is a 5 BP stem. The tRNA 5 to 3' structure contains the undercodon but in reverse order. The T-arm is a 4 to 5 BP stem containing the sequence, while it's pseudo-running, a modified uridine. Each subunit consists of 1 or more ribosome RNA molecules and a variety of ribosome proteins. The ribosomes are millets particles consist of RNA and associated proteins that function to synthesize proteins. The ribosome can be found floating within the cytoplasm or attached to the endoplasmic reticulum. This table shows the difference of the ribosome between bacteria and the mammalian cells. the mRNA carrying the DNA instruction and the tRNA carrying amino acid meets the ribosome. This cartoon shows the process of how it happens. The amino acids are joined together to make a protein. This cartoon shows the process how it happens. The ribosome joins the two amino acids and breaks the bond between the methionine and its tRNA. The tRNA flows away from the ribosome and following the ribosome to bind another tRNA. The ribosome moves along the mRNA, binding new tRNA molecules and amino acid. When ribosome reaches the stop codon, the whole process finishes to complete polypeptide. The initiation of protein synthesis. The protein synthetic machinery must select appropriate starting the points for mRNA reading and peptide bond formation. The start signal for protein synthesis is AUG. Starting signals are required for the initiation of protein synthesis in prokaryotes and eukaryotes. The 43S pre-initiation complex is assembled by association of ternary complex with the 40S ribosome subunit, to which initiation factor EIF1A and EIF3 are already bound. This step is followed by the binding of mRNA to form a 48S pre-initiation complex, a process that usually requires initiation factor EIF4E, 4G, 4A, and 4B. The rule of EIF4E is to bind to the 5' structure and the mRNA and to associate with EIF4G. The EIF4G also binds to EIF3, thus forming a bridge between the mRNA and the 40S ribosome subunit. Addition of the 60S ribosome subunit in a step requiring initiation factor EIF5 results in hydrolysis of the GTP associated with EIF2 and the loss of EIF3 from the ribosome. The 8S initiation complex thus formed and then competent to begin protein synthesis. Before EIF2 can be reutilized for another round of initiation, the GTP bound to it must be exchanged for new molecules of GTP in a reaction catalyzed by EIF2B. The phosphorylation of EIF2 on its subunit, position serine 51, blocks this exchange process and leads to the formation of a stable and inactive complex. The initiation of translation usually involves the interaction of certain key proteins and then the initiation factor with a special tag binds to 5' end of the mRNA molecules. The 5' cap as well as with this 5' untranslated region. These proteins bind to the small ribosome subunit and hold the mRNA in place. EIF3 is associated with the 40S ribosome subunit and plays a role in keeping the large ribosome subunit from prematurely binding. EIF3 also interacts with EIF4F complex, which consists of 3 other initiation factors, EIF4A, EIF4E, and EIF4G. EIF4G is a scaffolding protein that directly associates with both EIF3 and the other 2 complexes. The EIF4E is a capping binding protein bound of the cap by EIF4E is often considered the rate limiting step of capping dependent initiation. 43 pre-initiation complex accompanied by the protein factors move along to the mRNA chain towards to a 3' end. in the process known as scanning to reach the start codon, typically AUG. In eukaryotes and Archaea, the amino acid encodes by the start codon is methionine. What differentiates cap-independent translation from cap-dependent translation is that cap-independent translation does not require the 5' cap to initiate scanning from 5' end of the mRNA until the start codon. The ribosome can be trafficked to the start site by just direct binding initiation factors and or ITAFs, bypassing the leads to scan the entire 5' untranslated region. This method of translation has been found important in conditions that require the translation of specific mRNA during cell stress, when overall translation is reduced. Translation elongation is simply the ribosome traveling down the message, reading the codons and bringing in the proper amino tRNA to translate the mRNA out to the proteins. Eukaryotic elongation factor 1F, which is blue circled in this figure, is in complex with the GTP, which is labeled by red star, delivers a long acylated tRNA to the I site of the ribosome. The GTP is hydrolyzed and when the codon-antacodon recognition occurs, and the EF2A and the GTP is released from the ribosome. The GTP exchange factor for EIF1A is complex with 3 subunits. EEF1B is shown in orange, EEF1G is in green, and the EEF1D is shown in purple. So this complex promotes the exchange of the bound GDP for GTP to regenerate active EIF1 GTP. The ribosome reaches a stop codon on mRNA. This causes the release factor that promotes hydrolysis. So then finally, the translation complex deconstructed and moved to a larger translation circle. So here is the summary of translation central dogma. Here is the genetic information flow. The DNA transcribed to produce mRNA, the mRNA serves as a template to make proteins through translation machinery. The proteins are composed of amino acids. There are 20 different amino acids. The different proteins are made by combining these 20 amino acids in different combinations. That's all for chapter 4. Thanks for your attention. And here is some reference.